Let's get together to co-create a breakthrough for freedom of energy, sustenance, healing, and regeneration of the human vessel. Welcome to FESIG, the Free Energy Special Interest Group, where science meets spirituality in the quest for truth and knowledge to free humanity and transform this planet into a paradise.
Thank you, Pontus, for the intro. Hi, everyone. Welcome to FESIG's 92nd meeting. Today is the 8th of September. My apologies if some of you were trying to get into the meeting on the 1st of September. I postponed it to a week later. Not the first Wednesday of the month this month, but the second. It's because I had to prepare the meeting and I had a new laptop because my old laptop died on me. Anyway, thank you so much for taking time off to attend this meeting where we have some of the most distinguished speakers on the planet speak today, and it's so wonderful to have all of you here with us and the speakers as well. I would like to introduce the speaker, but first and foremost, I want to show the agenda of the meeting today. And just if you could help me share screen on the agenda, that'd be cool. Yeah. Okay, I'm Crystal Gore, the founder and initiator of FESIG. And I have the co-chairs with me, Pontus Habsjer. The other co-chair, Fraser's not here today, right? Yeah, and James, Ease and Eva, so Pontus, you co-chair with me. Pontus is the head of R&D. Fres is the chair of the R&D, which we hold meetings every Monday for 4 to 5 hours, yeah, because we got so much to do, to learn and to develop. So today we have, if you could scroll down to the next slide and the next slide and the next slide, we have the agenda. Yeah, I have to speed it up a bit because Jerry doesn't have much time left. Next one. Yeah, go to our website, the Free Energy Special Interest Group, to look at what we are about and learn about what we do. And then come and join us like the first Wednesday of every month. We have our meetings. Yes, that one, please. Onto the agenda. We are a platform where science meets spirituality, and we encourage scientists to go within themselves to solve the equation, because it is going within that is going to give them the eureka moment, right? So the first session today, the 92nd FESIG meeting, we have our honorable and distinguished speaker, Professor Dr. Gerald Pollack. He's talking about the fourth phase of water beyond solid, liquid, and vapor. Filling in the cells of your body, it is charged. The fourth phase is filling in the cells of your body, and it is charged, creating a battery that can produce electrical current. This talk will consider the potentially broad implications of this phase, not only for nature and spirituality, but also for health, with a better understanding how cells function. Wow, it's so exciting. And then once we've done the Q&A session, we move to the second session, where the successor of the late Dr. Emoto Masaru, Michiko Hayashi, who's the ambassador and global director of the Emoto Peace Project in Japan, will be talking about Emoto's famous work on the hidden messages and healing power of water, and his groundbreaking research and discovery that water retains all information and is deeply connected to our individual and collective consciousness. And after the Q&A session, then we will adjourn the 92nd meeting to the 93rd on the 6th of October. Please pen it down in the diary so you can join us again. And you can write to me at crystal@truevisionofpeace.com if you have some ideas that we can improve ourselves or whatever that you want to do. If you're attracted to our cause, then do join us and volunteer to help out or pay forward. That would be very much appreciated. So our website is truevisionpeace.com/FESIG.html. Thank you, Pontus, for helping me share screen here. I would like to introduce the speaker now, our distinguished speaker, Jerry Pollack is a scientist recognized worldwide as a dynamic speaker and author whose passion lies in plumbing the depths of natural truths, which is really what I really am admiring about him, you know, and we want him to succeed because we are truth seekers. And so truth seekers want truth out to the public as much as possible. Thank you so much for being such a brave warrior there, Jerry. Gerry Pollack received the first Emoto Peace Prize in 2016 and is a recipient of the University of Washington's highest honor, the Annual Faculty Lecturer Award. He is founding editor-in-chief of the research journal Water and director of the Institute for Venture Science. Dr. Pollack's award-winning books include The Fourth Phase of Water, 2013, and Cells, Gels, and the Engines of Life, 2001. His interests have ranged broadly from biological motion and cell biology to the interaction of biological surfaces with aqueous solutions. His 1990 book, Muscles and Molecules, Uncovering the Principles of Biological Motion, won an Excellence Award from the Society for Technical Communication. His 2001 book, Cells, Gels, and Engines of Life, and his newest book, The Fourth Phase of Water, Beyond Solid, Liquid, and Vapor, won that Society's Distinguished Award, their highest distinction. The later book went on to receive the World Summit Excellence Award. So you see, we have a very distinguished, accomplished scientist here with us. Pollack is now Professor of Bioengineering at the University of Washington faculty. He is convener of the Annual Conference on the Physics, Chemistry, and Biology of Water, and Executive Director of the Institute for Venture Science. He received the Biomedical Engineering Society's Distinguished Lecturer Award in year 2002. In 2008, his colleagues chose him as the recipient of his university's highest annual distinction, the UW Faculty Lecturer Award. Amongst the many distinguished awards he received, which you can read about in our FESIG website, in 2015, he won the BrandLaureate Award, previously bestowed on notables such as Nelson Mandela, Hillary Clinton, and Steve Jobs, and more recently, the Lifetime Achievement Award for the Chapel Natural Philosophy Society. He appears briefly in the 2016 Travis Rice sports action film, The Fourth Phase, named after his recent book. And he is included in the 2019 listing OOOM Magazine as one of the world's 100 most inspiring people. Finally, in early 2020, he presented his work at the Majlis by invitation from the Crown Prince of Abu Dhabi at his royal palace. Thank you, Jerry. Over to you now.
Well, thank you. Okay, well, I appreciate the kind introduction. So let me proceed with my talk. I share a screen. Let me do that. Whoops, what did I just fail to do? Here we go. Okay, so as you remarked, I want to talk about a different newly discovered phase of water, and how it may play a role in health and agriculture and life in general. And I'm going to be focusing on light and how light interacts with the water, a topic that for some of you may be completely new. When you speak about water, a lot of people think that there are no remaining questions. After all, you know, water has been studied for centuries by scientists, and it's a simple molecule, and everybody presumes that everything there is to know about water must already be known. I want to start by demonstrating to you that that's completely false, and let me show you how. We start with a common observation: the cloud sitting above the water. Clouds are made of water, and the question is, if it's made of water, you'd expect that if you were to reach up there with a pail of water and turn over the pail, the water would come down like a waterfall, but the cloud stays up. So why do clouds float? And if you don't know the answer, then you don't know all there is to know about water. Here's another one. 2 beakers, lip to lip, filled with water. Stick one electrode into one electrode into the other, and then see what happens. So the first thing that happens when you do that is a bridge forms between the two. What's more interesting is that if you move one beaker away from the other using a motorized stage, that bridge, so-called water bridge, persists. And it persists indefinitely for distances up to a few centimeters. How do you explain this? If you can't explain it, you don't know everything there is to know about water. Here's another one. This is a lizard from Central America, and it spends a lot of time sitting on branches. When it gets inspired, for reasons that nobody really knows, it jumps off that branch and jumps into the water, and it walks on water. And because it walks on water, it's known as a so-called Jesus Christ lizard, for walking on water. Well, how do you explain that? And if you don't know the answer, then you don't know all there is to know about water. And here's yet another one. This is a big magnet, this blue gadget here. With it's a superconducting magnet that generates high magnetic field. And in front of it is just a simple trough containing water. And a little red dye is thrown in for effect, but it's basically water. You turn on the magnet, and the red sea splits, as you see here, in such a way that Moses could walk across it. How do you explain that? So, here we go. All that you expect is known about water. Forget it. There is a lot that's unknown about that simple molecule. Well, I got my inspiration. We were in a different field studying muscles and muscle contraction from a guy named Gilbert Ling, who came from China. He was chosen among all of Chinese scholars right after World War II, the first group. There were 3 of them. One of them won a Nobel Prize. Some think he should have won 2 Nobel Prizes. Anyway, he passed recently, just shy of 100 years old. And one of his major contributions was about biological water. He said that biological water, I'm looking for my water, but I can't find it. Biological water was not like water in a glass. He said, in water in a glass, the molecules are randomly oriented and bouncing around a huge number of times per second. He said, no. Inside the cell, water is different. It actually, the water, each molecule stacks upon another molecule, giving you something like soldiers at attention. That was his point of view, and he had a lot of evidence to suggest that. I met him at a conference more than 20 years ago in Hungary, and not only was I inspired by him and his presentation and discussions, but also by a dozen other people who came to present evidence that the water in the cell really did differ from ordinary liquid water in a glass. And so I was really amazed, and I came back home to Seattle thinking that this is so important that I should do something. We should change our research direction. And I gave one of his books to some of my students to look at, and each one of them came back to me with a similar sentiment, namely that this guy is onto something really important. And if he's right, this changes all of biology, all of cell biology, because water is central. And so I decided first thing is to write a book about it, because Gilbert Ling's books are challenging to read. And so I undertook that, and in 2001, that's just 20 years ago, I wrote this book, and it received mixed reviews. Some said, "Oh, this is just more of Gilbert Ling, and we know that he's a crackpot, so pay no attention to it." 2 others, like someone from Harvard University, who said this is a 305-page preface to the future of cell biology. Well, you know, among thoughtful readers, they'll pick this book out of many other choices, and well, this one seems to be pretty intent on reading. 1 year later, still reading the book. So I'm pleased by that. Well, if you want to illustrate Gilbert Ling's central idea, it looks something like this. So inside the cell are solids, and the most populous solids are proteins, and proteins generally have charges on their surfaces, as you see here, and these little bean-like things represent water molecules, which are in fact dipoles. So this dipole then has plus here and minus here, and the plus will be attracted to the minus, you see, and stick, and the opposite here, and so on. And so you'll get these water molecules to line up this way. And mainstream physical chemists say, well, yeah, sure, of course, there may be one or two of these ordered layers of water molecules, but beyond that, they begin to disorder because of the effect of Brownian motion or thermal motion, which means that molecules tend to jitter, to jump around. And Gilbert said, well, yeah, that is true, but the evidence we have suggests that these ordered molecules can actually line up dozens, if not hundreds of layers, which would be a radical change in terms of cell biology. And so we actually looked for an experimental model that could demonstrate this. And one clue is that when you have order like this, it's like a crystal. And crystals, as they form, they tend to exclude solutes and particles. You see, just like ice, you know, when ice forms, the water that goes into the formation of ice may have particles and such dissolved, but as the crystal forms, it pushes them out. You see, so you get a pure crystal. If it's pure, it can't contain contaminants or other molecules. So that's what we were looking for, some example of a place where you'd expect solutes to be or particles to be, but they're not. And we found it. So we put a gel, the edge of which is shown here. Sorry for the focus issue. This is one of the first images. And we put it into a chamber, and into the chamber, we put water plus particles. And these particles were little spheres called microspheres. And when we looked in the microscope, we could see immediately that there was a region here that contained none of these spheres. And we thought, oh, this looks pretty interesting. You know, maybe this is a region where the water molecules line up and push these out. And the phenomenon is actually more prominent than I show here. If you look at the video, you could see this zone here, it seems to be building. This is over a period of about 3 or 4 minutes and pushing these particles out. And we began to see this again and again, and we're scratching our heads wondering if this huge region, this is about half the thickness of a human hair, might actually be a region where the water molecules are lined up. This is a pretty big region. We kept seeing it, and the Australian colleague said we should call it something because, you know, it's awkward to go through a long speech to explain this. And he suggested call it exclusion zone because it excludes. And in the US, it's really convenient because exclusion zone is EZ. In England and many other places, unfortunately, it's EZ instead of EZ. So being easy to remember doesn't work over there. Anyway, we call this EZ, and later we began calling it fourth phase because everything about it is different. So in terms of generality, we did many experiments, and to summarize, because I have limited time, many of these charged or hydrophilic surfaces generate exclusion zones, and many solutes are excluded. So it's very, very general. In fact, I show one in plants. You can see this is xylem. I forget the species, but you can see, you know, these tubes that run from the bottom to the top of the tall tree. You can see that the microspheres put in here and examined by electron microscopy by one of the greats in the field. There are very few microspheres here at the edges, but plenty here, suggesting that indeed that in the xylem there is EZ, as in many other places. So the question, though, is this water really different from bulk water, from ordinary water? And I've alluded to the possibility, but I presented no evidence. So I just want to give you, in a sort of flash, some of the evidence. So this is evidence that exclusion zone or EZ water is physically different from ordinary bulk water. And the reds represent the method that we used, and in black are the observations. And I'm not going to go through all of these, just one of them, because I'll take up all the time that we have. But just to give you an idea, you know, with nuclear magnetic resonance, we found that the EZ water molecules were more constrained than ordinary water molecules, and so on. And this list was constructed about 10 years ago. And since then, probably if I constructed an up-to-date list, there would be almost double this size. So there's a lot of evidence that this EZ water differs substantially, if not completely, from ordinary liquid water. I just want to mention this one, the negative charge, because it's critically important for what follows. And we did that, we measured that directly in a laboratory preparation using micro electrodes, little electrodes, invented, by the way, by Gilbert Ling, the same guy, to measure the electrical potential. And we did many such measurements, and all published such, and it indicated something really, really interesting. And that is that the EZ water, this is, by the way, the surface, it could be a gel surface or any water-loving or hydrophilic surface sitting next to water. What happens is that the EZ grows, develops over just a few minutes, and is negatively charged. We found that by sticking electrodes directly here, and the same, finding that this is equally positive charge here. And the way that happened, and this is like a battery, right? Separated plus and minus. And the way that happens is it all starts with water molecules, because, you know, initially these were just water molecules, and energy comes in. I'll describe the kind of energy in a moment, to the water molecule, and it splits the water molecule here or here, and you get a plus hydrogen here, and you get a minus because this has a valence, usually -2 + 1, giving you a net of -1. And so these are the entities that gather together to form this zone, and it's negatively charged. And these are cast out of this zone into here, which gives them a positive charge. So this is more or less how it forms from ordinary water undergoing a radical change to EZ water with negative charge. And if you look at the detailed structure, which we could deduce from many experiments, we have this material, the hydrophilic water-loving material with charges on its surface. And from the water that's sitting next to it, these EZ layers form, one and then another one and another one and so on. And they keep forming, as shown, from the water. And as you can see, the structure, which we could deduce, is radically different from ordinary water. And if you were to look at it from the point of view looking at the surface, it looks like this is a honeycomb structure consisting of oxygens at the vertices and hydrogens here. And if you were to count, for example, the number of oxygens and hydrogens in a single unit cell, you'd find that it's not H2O. And you wouldn't expect it to be H2O, because H2O is neutral. And as I mentioned to you, the experimental results dictate that this is negative, not neutral. And therefore, uh, if you do the actual count, it turns out it's H3O2. If you had H4O2, it would be double this exactly, and it would be neutral. But it's not neutral because this is missing one positive charge. So it's got net negative charges, which again is what the experiments dictate. So this is the basic structure that we could deduce. And I asked the question now about, which I think is of interest to your group, but maybe in a more practical way. Could EZ water contain information? And whoops, why is this not moving? What have I done wrong? Okay. We know about information. This is a radical idea that water could contain information, but we know about information and the principles that are required from looking at one of these thumb drives, which I think most of you are familiar with. It basically contains memory inside. And what are the distinguishing features of this memory? Well, the memory is generally built of silicon, built of transistors. So the transistors are laid out here in a planar fashion in a very regularly disposed way. This is like a crystal lattice. So that's the first requirement. Second requirement is that each of these transistors can undergo a change from a change of state, if you will, from 0 to 1 or on or off, or however you want to describe it. So those are the two main features. And we asked the question, does EZ water contain similar features? And that's a necessary condition for it to store information. And as you can see here, it does exactly. It consists of oxygens and hydrogens all laid out in a very regular array, and actually 3 dimensions rather than, in fact, 2 dimensions. So first requirement is met. Second requirement is that either the oxygen or the hydrogen needs to have at least 2 states. It turns out oxygen has 5 states. You find this in any chemistry book. The most familiar state is the -2 state. We call it valence of -2. And then, but there's also -1, 0, +1, and +2. So it satisfies that second requirement as well in spades, even more than we might expect. And so yes, it is possible that water, or particularly EZ water, contain information. And the person most responsible for all of this, and I know that Michiko Hayashi in the next presentation, who learned from Dr. Emoto, shows or will explain his results in more detail. But Emoto put his intention to the water and also exposed it to, or could expose it to, physical kinds of attributes, and and froze it. And the ice crystal shape depended on his intention. And so here are just some examples. So it starts with sound. A Mozart symphony, play it to water, freeze it. And here's a pretty crystal. John Lennon, Imagine, also pretty crystal. And then just thinking about peace or thank you resulted when frozen, the water is frozen in beautiful crystals. You Fool gave rise to something rather ugly, and heavy metal music, the same. You'll hear more about this, but this is the result of Emoto. And Emoto's work is not well regarded among scientists because of the way he chose his results. So he would freeze, say, 50 Petri dishes that have been exposed to intention, and then pick out the one that illustrated best what he wanted to illustrate. And that's not a particularly scientific way of doing it. On the other hand, the people from the Emoto group are now attempting to do it in a more scientific way. So, and the idea of water memory is not unique to the Emoto group, but in fact, it's attracted people like Nobel laureate Luc Montagnier, who has done, you might say, controversial experiments that actually indicate that water information or memory can occur inside our bodies, and our bodies are filled with easy water, which I'll go into in a moment. So it's not a surprise if memory is stored in easy water that you can communicate subtle kinds of information that may be stored in the water. I just want to interject something here, a recent result that may play a role in practicality of all this, and that is, excuse me, that easy water can be solidified at room temperature. Now, you all know about solid water, that's ice, but ice is formed at low temperature. But we're talking now about a kind of water that can be a solid at room temperature. This is really a shocker. And it's not from our laboratory, at least originally, but so once again, why is this failing to advance? But by Vittorio Elia in Naples, who has published several papers on this, solidifying a kind of water that was initially a liquid or kind of gooey like honey, and making it a solid at room temperature. And this demonstrates the solid that it's very scarce, difficult to make, but when you make it, and we've duplicated that in our laboratory, and we're about to send it in for publication. So, and we know that there are no contaminants in the water from our experiments that could have caused anything like this. So this is really pure, easy water that's a solid at room temperature. And so you can imagine that the idea of putting easy water in here, it might be a little bit awkward to put a liquid inside, but since it could be made into a solid, which lasts indefinitely, it's possible to put easy water in here to constitute a real practical water memory with solid easy water. And we computed that because the easy memory capacity is at the atomic level rather than at the transistor level, and because there are 5 states rather than 2 states, you can pack in huge amounts of information. We calculated that it's up to 1 billion times of what a current unit can accommodate. So we're really excited about this possibility. There's a lot of work to be done to demonstrate its practicality. But another point is that the same thing occurs inside your cells. So the cells are crowded with solids. This is a gross underestimate. And because it's so crowded, all the water is sitting, essentially all the water is sitting near a protein or another solid like nucleic acid. And therefore, since these are charged hydrophilic surfaces, essentially all the water inside your cell is EZ water. And therefore, if it's EZ water, it's subject to the influence of these kinds of subtle energies that, that people have been demonstrating can affect water and can affect us. So these people, healthy looking people, are absolutely filled with EZ water. Their cells, and even outside their cells, are filled with EZ water. And therefore, any kind of subtle information that is sent to them and received by them can be received biologically. And, you know, it's the kind of thing you walk into a room and you see somebody and there's a kind of, besides the verbal information, there's subtle information, something that you feel or you sense, but you don't know exactly where it's coming from. Could be EZ water that fills the cells of these healthy people. And some of the rest of us, so long as we remain healthy. I'll discuss that in a bit. So we return. The original question in this section was, is EZ water physically distinct from bulk water? And I mentioned that briefly, and the answer, I think, is yes. The evidence that we have is a layered, sheet-like honeycomb structure. It grows layer by layer that has information storage capability. So very exciting. Now, I mentioned early on that the EZ water was like a battery, if you remember. And all batteries need to be charged. So if you forget to connect your cell phone or smartphone, you go to sleep in the morning, it won't be working. You don't have any power. And it goes for all batteries. You need to charge them. And so what we found is that this charging occurs from incident radiant energy or light. This was a surprise. We had scratched our heads for a long time and couldn't figure out where the energy was coming from. But actually, it was an undergraduate student who found it. The student was doing what he was not supposed to do. And I tend to encourage that. And what he found, and this is a picture, actually, one of the early pictures coming from him. This is obviously not what he used, but he found a gooseneck lamp sitting right next to him on the bench. And he turned it on and he shined it on the preparation. And the region that received the light, you can see what happened. So typically, if you have a hydrophilic material, in his case, it's Naphion, which is a polymer that we use, it's convenient. And typically, this exclusion zone would grow and the microspheres would be outside here. And the boundary between the EZ and the ordinary water with the microspheres is usually parallel, as it is here, to this surface. It grows sheet by sheet. However, where he was shining the light, it grew rather substantially. And so he called me in and I saw this and I thought, my goodness, this student in his naive way figured out the answer. Where does the energy come from? Well, it looked like it was coming from light. The photons of light on here were growing the exclusion zone. So, it didn't take a rocket scientist to figure out that this is what's going on, that light, photons are responsible, the energy from photons are responsible for building the exclusion zone. Simple answer. But naturally, from an anecdotal observation like this, we wanted to find out more, and we did experiments. We wanted to find out which wavelengths of light are most important. So we looked at the visible spectrum of light. The spectrum extends beyond the visible into the ultraviolet at the short wavelength and infrared at the long wavelength. And to make a long story short, most of it was irrelevant, did nothing except for infrared. That was the most effective. And infrared, a lot of people don't know about infrared energy. They think, oh yeah, turn on the electric range, look inside and it's hot and you can see glowing coils like in a toaster. And there's infrared energy and radiant energy and heat. And they think, many people think that that's it. But it turns out that everything is generating infrared energy all around you. And the way you could detect it is you take a camera, just like your smartphone camera, but the sensor inside the camera is sensitive not to visible light, but to infrared light, slightly longer wavelength. And even if it's dark, you turn off all the lights in your room and looked with your infrared sensor, you get a beautiful image of everything. Even Crystal, on that dress that you're wearing and all the nice colors that are shining through, you'd see images of all of that. And so since this energy is all over the place, you have to go to incredible extremes to eliminate it. It means that the energy for building EZ water is available. Once again, is available. It means that if you have a hydrophilic water-loving material sitting next to water, because the energy is always there, you'll have some EZ water. And if you add to that ambient energy, more energy like from that lamp that you saw, then the EZ water will build. EZ water will build. And if you take it away, it will return back to its default level. So infrared level is really, infrared energy is critical for this buildup. So I asked the question earlier, what is the energy for buildup? And the answer is light. Photonic energy is responsible for building the EZ. And that energy orders the water, because you need energy to create order. That's a fundamental theme of thermodynamics. And it also charges the water battery. It separates that charge and gives us energy. So that's the energy we're talking about. And this sort of illustrates it. I gave a talk recently to Israel. So there are nice beaches in Tel Aviv, but it could be Hawaii, or it could be whatever you like. And maybe even London and in this heat. So the idea is that you've got this water exposed to sunlight. And mind you, sunlight is roughly 50% infrared energy, even more than 50%. So the energy that reaches you as heat comes directly from the sun. This builds EZ water. And so all you need to do is essentially lie in the sun, or the water needs to lie in the sun, and EZ water gets built from the sun. And if that's the case, then those of you who are paying attention may think, well, gee, if this gets full of energy, you ought to be able to harvest this energy in some way and use it for, constructive purposes. On the other hand, I bet not anybody who is watching or listening has ever had the experience of seeing a glass of water doing work, right? I think so. But I'm going to demonstrate to you that a glass of water can indeed do work. And let me start with this observation by yet another clever undergraduate student working in my lab. In the past, we've had many. And so Arthur was responsible. His project was to take Naphion, and we had just found that Naphion comes not only in sheet-like, a polymer in sheet-like form, but also in tube-like form, like a straw. And we were wondering whether the curvature might impair the buildup of EZ water next to the surface. So I asked him, please look here, just outside and just inside, and see if there's an exclusion zone. And he did, and he found that within 1 or 2 weeks, it was really easy. But then I didn't have enough time to connect with him, so he went about doing his own thing. And his own thing actually amounted to something more interesting than what we had found. And so what he did was, well, he told me, he came running into my office one day. I was sitting there with some important visitor, and he came barging in. Usually the students don't come barging in. They come in a little bit timidly. They'll kind of push the door open or something like this and say, are you busy or whatever? But he's just barging. He said, I found something interesting I had to tell you. And my visitor, I think, was not memorable. I can't remember who it was, but possibly some important person. And Arthur came and said, here's what I found. He said, I'm looking and I can see water flowing through the tube and flowing through without stopping. And I thought, my goodness, if Arthur is right, this is amazing, because you need energy to push the water through the tube. It's sort of like you need the heart, and more maybe, you'll see in a moment, to build up pressure to drive the water. And there's no pressure difference between here and here, because the tube is sitting horizontally at the bottom of the chamber, and the pressure here is the same as here. So something else, some other energy must be responsible. And I'm thinking, if he's right, the energy we're talking about is probably what we found before, the infrared energy that's getting absorbed and converted into other kinds of energy. So we checked carefully, and Arthur was absolutely right about what he was finding. And so, and this led to a lot of really, really interesting stuff. So first, this is how the experiment is done. You take a tube of Nafion, inside you put water plus microspheres, and make sure there are no bubbles, and then put it into water plus microspheres, and look at it in the microscope under low light. We don't want the light to, so we use just green light, get rid of everything else. And here's what you see. Here's the Nafion tube, the exclusion zone right next to it, and the flow of water with microspheres through it. I stopped the video, but if you, we found in the laboratory that this could go on for at least a day or a day and a half, essentially indefinitely. And we know what to do to actually make it persist from that. Well, we showed it to quite a few people, and one astute person said, "Hey, I'm not sure that this is right, and maybe there's something really weird about Nafion, and why don't you try some other tubes and see if you get the same result to demonstrate that it's not a special unexplained feature of Nafion." Well, we couldn't find the right tubes, and so we made our own. Essentially, one of the students in the lab took a gel, and the gel, as it forms, it goes from liquid to solid. He stuck a wire in it, and just as it was gelling, he pulled out the wire. So we wound up with a little chunk of gel with a tunnel running through it. We stuck that in the water with the microspheres, and essentially we had the same. And in this case, instead of Nafion, it was a gel that was sitting here. And here's an example. So it's a polyacrylic acid gel. You see it here, and you see it here. And next to the gel, after a few minutes, EZ built, as I've shown you before, EZ built that I've shown, pushing the microspheres to this central region. If you look at the video, you can see that the same thing occurs. And we've done this now with 7 or 8 different gels. Same result in each case, although the velocity is different and the EZ size is different, but the phenomena is the same. So this is not, it's general. It's not a feature of Nafion alone. And somebody else came and said, well, you really think this is driven by light energy? Why don't you crank up the microscope light and see if it goes faster? We did that. We published the result. We could get up to a 5 times speed increase just by cranking up the microscope light. So this is run by light. And so, we concluded that this is interesting, that we put a hollow tube inside of water, and you can see the flow through it is essentially indefinite. Work is done, and if work is done, you need energy to do the work. And in the absence of any other energy mechanism that we or others have found, the absorption of infrared light is a necessary condition. And so therefore, we found with this energy that water becomes a transducer of light energy, transducing it into, you might say, mechanical energy of flow. So a glass of water can do work. It's not unreasonable. And the central agent is EZ. So a question arises, this is an interesting principle because you can get energy from the environment. And if you can get energy from the environment, the question arises, does human biology use radiant energy? Now, if you were Mother Nature, this is the best image that I could get of that mother. She looks maybe too young to be a mother, but Mother Nature. So Mother Nature invented single-celled organisms and green plants. And then one day, she decided, I'm getting bored. I think I will invent animals. And animals have the capacity to forage, find food, eat plants, and they can get their energy that way. But if you were Mother Nature and you were so successful using light in all of those more primitive species, would you throw that away, cast it out, and say, forget it, I'm going to start all over? Or would you keep it in reserve as a kind of supplementary entity just in case, or even to supplement or complement the usual energy mechanism through food, you're getting a lot of infrared energy. It's actually not just from outside, but the heat that you generate from inside also is generating infrared energy. So you're getting a lot of it. And when you consider how we might conceivably use some of this light or infrared energy, the first thing that may come to mind is the cardiovascular system. And you've got a lot of capillaries sitting, for example, near the surface. The infrared light penetrates and maybe has something to do with assisting the heart in propelling the flow, maybe. So at first, this was a kind of radical speculation. And then I took a trip to Moscow, and I had occasion to visit the laboratory of my dear friend, Vladimir Vyakov at Moscow University Biochemistry Department. And he quickly introduced me to his colleague from the laboratory. And his colleague told me, he said, through the translation, he said, there's a problem in the cardiovascular system. And I got to admit, I was skeptical. He said, it's a big problem. That increases my skepticism because as a graduate student, I did computer simulations of what happens, primitive computers at the time, what happens in the cardiovascular system, the pressures, the flows and such. And I thought we had it nailed, that we really understood it very, very well. And he started to tell me, no, we don't understand it very well because there's a big problem. Okay, tell me the problem. He said, the problem is this, the capillaries are narrow. In healthy young adults, the capillaries might be only 3 or 4 micrometers in diameter. But the red blood cells that need to pass through those narrow vessels is twice the diameter, 6 or 7 micrometers. So, we assume that nature didn't screw up or something. These red blood cells, erythrocytes, have to make their way through. And in order to squeeze through, it requires a lot of energy. And they calculated the amount of energy, and they came to the conclusion that if the heart alone were responsible for this, the heart would need to generate a pressure that is something like 1 million times the actual pressure that it generates. You might call that high blood pressure. So they said there's got to be another source of energy somewhere, and they speculated about a couple of possibilities. So I just want to show you. So this is what red blood cells look like in general, if you just look at them, but not inside the vessel. And here you can look about inside the vessel. This is a piece of muscle tissue. You can see the striations in it. And here are capillaries running here and here. And you can see that as those red blood cells go through the capillaries, they get squeezed. And you need a lot of energy to accomplish that squeeze, much more than the heart could ever develop. And it looks, you can see it also by video. You can see this guy is having a really hard time. A lot of energy is needed to compress them in order that they can pass through. So this guy is speculating on what the mechanisms were, and I'm thinking we just found something in the laboratory that might be the answer. We found that hollow tubes in water will generate flow through the EZ that's actually inside, and it's radiant energy that actually drives the flow. So the question rose to me, well, is it possible that radiant energy might also drive blood flow in our vessels? In other words, the same mechanism could occur inside of you and me. We actually saw something in literature. This is my student, Zheng Li, who actually did the experiments I'm going to tell you about. And he found that there was a paper published about a decade ago from an Israeli group who studied mice, and they were studying blood flow. And after their experiment, they sacrificed the mouse. The mice died within 10 seconds, but flow continued. And they were astonished to find that without the heart beating, flow continued. Then they looked and they studied it for 10 minutes, 20 minutes, 30 minutes, 1 hour, and the flow continued. Lower velocity, but it continued. And they repeated the experiment, 10 mice, they got the same result each time, and they published it. What they hadn't realized, in fact, was that a half dozen different groups over the past century or so published similar results. Heart stops in different contexts. Heart stops, flow continues, lower velocity. But something besides the heart then has to be driving the flow. So we did experiments to test, and we tested the central feature, the signature feature of this mechanism, namely that infrared energy was responsible. So what Lee did, he started with a fertilized egg grown for 3 days beyond fertilization. He lopped off the top of the egg, and so you can see the cardiovascular system, developed an imaging system to look at representative vessels. And the first thing he did was to stop the heart by injecting potassium chloride, and the heart stopped. And he wanted to see if the flow continued. And he found, indeed, that the flow continued. So here is his result. This is blood velocity versus time. So the first thing is you stop the heart, and the flow goes down, considerably down, but it doesn't go to zero. Some flow is maintained. And then he tested that signature feature by exposing this to an infrared lamp. And you can see what happened is that the flow increased by a factor of about 3 times. And then he stopped it, took the lamp away, and the flow went back down to the original. So it's reversible. And indeed, the signature feature of the mechanism that we found in the laboratory appears in a living biological cardiovascular system. And that would include my system, your system, et cetera. And remember, we have infrared coming in from the outside and also from the inside because of metabolism generating heat. So this could be important and useful understanding that it's not just the heart, just our hearts that are powering the flow, because not enough energy. But inside the vessels themselves, there's a flow generator. And so radiant energy apparently does help drive blood flow. And similar in plants. Because we have not much time, let me just skip the plant business. And so more generally, how does radiant energy facilitate function? We start with primitive systems, green algae, photosynthesis, light is really important. Certain bacteria, light is really important. And green plants, which we know very well, it's also light. And here's, the rain falls, the roots absorb the water, which are then sent through the xylem to the leaves. And through the energy of sunlight, the very first step of photosynthesis is the breakup of water into H plus and OH minus. This is well established. It's 100% efficient, and it's in all plants, you see. And how does that relate to what I've shown? Well, remember, same thing happens. You've got hydrophilic material next to water, and the water is split into, as you remember, the negative OH minus, which forms the 4th phase, and the H plus, protons, which are generated out here. And so we have the splitting of water molecules into these two, very much the same as what we now know occurs as the first step of about 20 steps in photosynthesis. Very similar. And if you think this is really strange and odd, let me tell you that sorry, one slide. Anyway, the process is very similar. And when you think about what's going on in cell function, you've got a cell, and the water, EZ water forms because these solid surfaces are hydrophilic and got negative charge, and the cell is very crowded, and therefore this negative EZ practically fills the cell. And what happens is that the negative charges repel each other. They want to get out because they repel. And this is potential energy. They want to go from that high energy state to a low energy state. And when that does, that potential energy gets used to power the folding of proteins. Now, I know some of you are not so familiar with this, but it's generally accepted that when cells work, when they do their thing, they occur by protein folding. That is, the protein goes, say, from this state to this state, from an extended state to a contracted state. And the energy for doing that comes from this EZ water. And just to illustrate how it actually occurs, so here's a protein, and it's surrounded by EZ water, right? And so what happens is that when the cell goes into action, the EZ water melts. And as it melts into ordinary water, the protein undergoes a folding from the extended state to this contracted state. And then when the cell is finished doing its thing, for example, a muscle cell finished contracting, it returns to where it was, and the EZ water builds up again. That's the energy requiring state. That's normal. However, if you don't have EZ water, or you don't have enough EZ water, then it's different. It's impotent. It can't return to its initial state. And if it's a muscle cell, it'll remain contracted. And you've experienced that after heavy exercise. Sometimes your muscle is contracted up. So where does the energy come from? And as I began to argue, it comes from EZ. The way it works is, we know, just sort of summarizes some of what I've told you, light builds EZ, okay, infrared light mostly. That builds a negative charge. That contains the potential energy. And that potential energy goes into protein folding or work. It's the work cell. And if you connect the two, light is responsible for work of the cell. Now, obviously, it's not responsible for all of the work of the cell, but at least when we ask, where do we get our energy for doing work? Much of it comes from food, no doubt, but some of it, I will argue, comes from light energy, which is absorbed by the water, builds EZ, and gives us energy. And so should this matter to you? Well, yeah, it should matter to you, because light matters and water matters, and particularly infrared light. The two of them really do matter. And so what you want to do is, you want to address the question, for you to remain healthy, what is it that builds EZ water in your cells? Because you want that to happen. Well, first of all, it's water. And you need to drink a lot of water, because water is the raw material for building EZ water. Your body converts it into EZ water. A shortcut is green juicing. You go into your garden, you take some freshly grown plants, you squeeze it to extract the juice. What is that juice? Well, that juice is from the inside of plant cells, and it contains lots of EZ water. So you drink the EZ water, and you bypass the step that's necessary for building EZ water inside your body, because you're drinking it. And so many health practitioners report that green juicing is probably the best, least expensive, simplest thing you can do to promote health and reverse pathology. And it seems to work, at least the report I've gotten from many. Then there are substances that have been known since Ayurvedic times to promote health. We're talking 5,000 years ago, and used to this day, and they seem to be effective. We raise the question, could it be that the vast effectiveness, so many systems in your body that are impacted positively by many of these agents, come from their buildup of EZ water? We tested it, we confirmed it. Each one of these at concentrations that are normal to your body builds EZ water. And it includes turmeric, coconut water, basil, ghee, clarified butter especially, very strong, etc. Sunshine in Seattle is a bit like London, we don't get too much of it in the wintertime. You know, when the sun comes out, people feel good. And why do they feel good? Well, possibility is that they feel good uh, because the EZ water is penetrating, I'm sorry, the sunshine, the infrared energy from the sun is penetrating their body, including their brain. And so EZ water is built in your nerve cells inside your brain. Your brain is now functioning normally as it ought to, and your normal situation is feeling good, not feeling depressed or not feeling down, and the extreme of that is actually a sauna, or as the Finns say, sauna. And why is that? Well, it's heat, whether it's moist or dry. Heat is essentially infrared, not exactly the same, but heat is infrared energy. Infrared builds EZ. You take off your clothes, you expose yourself to infrared energy, builds EZ throughout your body, and you emerge feeling better. If you've had muscle aches, they're gone. If you have a headache, it's gone. If you're short of energy, you're revitalized. And another one that finally is grounding or earthing, I guess as you would call it. Why does that work? It works. There's lots of biophysical evidence that you connect yourself to the ground, like, for example, walking barefoot near the water on the beach. Why is that? Well, it turns out that I think the reason is very simple, is that the earth is negatively charged. We never learned that in the US. In Russia, I'm told by my friend and colleague that every middle school student knows it. And there's plenty of evidence for it. It's just that we have forgotten. So when you connect yourself electrically to the earth, you're connecting yourself to a practically infinite sea of negative charge. And that negative charge seeps into your body. And we know from our experiments, you add negative charge to water, it builds EZ. So I think the reason you feel better through grounding or earthing is simply that, adding negative charge to your body. So I end with practical applications. And the first is, everybody likes practical applications. First is getting energy from water and light. You've seen this enough times, you know it. But now you put electrodes inside, like so, and you'd expect that if you stick one electrode in the negative, one in the positive, this being like a battery, ought to be able to light a light bulb, right? And we demonstrate that you can. So this is a little primitive laboratory setup. You've got, I think, a dozen or so little chambers here, each one containing naphthalene, water, 2 electrodes, one in the negative region, one in the positive region, a switch here, a magnifier above light emitting diode, LED. And here's what happens. So you turn the switch and voila, it lights the bulb. So this is getting energy from light and water, entirely renewable. Whether it can work in a practical way, we think so, but sometimes, you know, it's not easy to go from a simple laboratory demonstration to practical application. You need to cross the valley of death, so to speak. And many laboratory inventions don't make it. We hope this will. Another one is getting drinking water from contaminated water. So it looks a little complicated, but the principle is very simple. We have a naphthalene tube or equivalent, similar to what I've shown you before. You know, it develops an exclusion zone here, and the flow comes into this tube, and the flow may contain contaminants like bacteria, viruses, microplastics, you name it. We use, as sample contaminants, we use microspheres, although we've tried viruses and bacteria with similar results. And you can see what happens. You get the exclusion zone here and the microspheres here. And then what we built is a differential extractor. And what this does is it collects the central, it's just 2 concentric tubes, and all the junk here gets collected by the central tubes and gets dumped here. And the region here, which is actually EZ water, comes and gets dumped here. And you can see that most of the microspheres are gone from here and present here. And we've been able to achieve as much as 200 to 1 separation. This is not 200 to 1, but we've been able to achieve that in a single pass. So in principle, it works really well. And we've been working to develop this into practicality. The real prize, again, we started to work on this, has to do with salt, with separating salt. So if you, for example, put water in here, salt water, thinking of salt as the contaminant, if you will, then the idea is that this would be the salt. You get the salt and you dump the salt, and you have water without salt, or very little salt, that you can use for drinking. So you can take ocean water and convert it into drinking water. We have some preliminary evidence that this might work, but a long way to go before making it a reality. And the beauty of this is that there's no physical filter. So you don't need to clean it or replace it. The filtration occurs by virtue of the energy from the sun, which separates, which builds the EZ, you see. And so the places where this is needed most, which would be, for example, the Middle East, North Africa, or so, there's plenty of sunlight, but they don't have much drinking water, so it fits ideally. So I reached the conclusion, I'm sorry this goes a little over time and I'm limiting the questions a bit, if there are any. So there are 4 phases of water, and the 4th phase is EZ phase. It turns out that this hexagonal structure of EZ is not too different from the hexagonal structure that appears in ice. And we did experiments to show that if you want to freeze water, you can't go from water to ice. You must go through this EZ phase. And if you melt the ice, you can't go from ice to water. You must go through this EZ phase. See, so that's why I put it here. It's integral to an understanding of what happens during freezing and during melting. But it is the 4th phase. Water has 4 phases, not 3 phases. And the implications of this are really broad. The central feature is that water is absorbing infrared energy and converting that infrared energy to useful work. I've given you an example of your cardiovascular system, which from our experiments, we conclude is driven not only by the heart, but also by the vessels. There are other biological systems, basically all cells. In chemistry, you've read about reactions from your maybe university chemistry class, or even high school. And all the reactions that you read about in water, not one of them deals with the presence of EZ or the role of infrared energy, light, charge separation. And therefore, if what I presented to you is true, or even part of it is true, many of the interpretations that appear in the chemistry books will need revision. When we hear about weather and think about weather, we think about pressure and temperature. Nobody, almost literally nobody, talks about charge. Clouds are charged, not only the thunder clouds, but every cloud. And this was demonstrated 60 or 70 years ago, but forgotten. I think that charge is absolutely essential for understanding of weather. And my next book, which should be coming fairly soon, describes how exactly. Water is important for health, as I indicated. You need to drink a lot of it to keep that EZ built, to retain health. If you want to process food in some ways, if you want to freeze it, or if you want to dehydrate, it's good to know something about those processes. And I think what I presented alludes to all of those processes. Practical issues, this can be used in filtration, and we've made some headway in that direction, possibly also for desalination and for getting electricity. So before my final slide, I would just mention something that the Institute for Venture Science, a new organization that we've been developing, which funds promising ideas that may bring radical advances or even scientific revolutions. We fund ideas that are radical. but that if they prove to be valid, will shake the earth. And we've identified out of more than 200 pre-proposals, from down to 15 proposals, we've selected 5 exciting projects which have the capacity to shake the earth if they prove valid. And we think there's a good chance that they may. We have some donations. We're looking for donors, people who have done well and want to give back to society in meaningful ways to promote things that can happen really quickly. And we think that this can happen in the Institute for Venture Science. So my final slide is the latest book. It's called The Fourth Phase of Water, and it describes much of what I've talked about, and plus a lot of other stuff that that is also relevant and associated with what I've been talking about. And it's a fun book, I think, to read. People have commented that you don't need to be an expert to read it, and it's full of cartoons and stuff like that. And so I recommend it if the material interests you. There's a lot more in the book. So I thank you very much, and I guess I'll stop here. And I have a few minutes. I'm sorry, we were delayed, and I have
thank you so much, Jerry. It's amazing,
my pleasure.
Fantastic, your presentation. We've learned
thank you.
so much from you. Gosh,
thank you so much.
I apologize that we started late. Otherwise, we would have a live Q&A session with you. I know you have to run because you have an appointment right now, and you're like 7 minutes off. I'm so sorry.
Yeah, well, it's okay. It's okay. I think I can handle one question if anybody has. If not,
Wonderful.
I'll just disappear.
Press, our R&D chair might have a question.
Thank you very much. When we look at the extraction of EZ water, you notice that you said that there was a time delay between the extraction and then it reverted back to the state of regular water. You alluded also to the green extract, and we know from those of us that have done fruit juices and that type of stuff, the faster you get it consumed in the line of from the time it's expressed out of the plant material, pressed out of it, the better it is. Is there other ways of extracting EZ water into another form that holds it in that state is the question.
Yes, and that is solidifying the fourth phase. And that's one of the reasons why we've been interested in that. I mentioned partway through that you can take EZ water and through a actually fairly simple process, have it become a solid. That solid lasts indefinitely. By indefinitely, we haven't done experiments that are indefinite, but essentially indefinite, as other solids would do. It's also true that under certain circumstances, and we haven't defined that exactly yet, the EZ water can persist for I would say indefinite, but substantial lengths of time. We haven't pinned down that yet exactly, but we know that if the EZ occurs in a large volume of water, it will persist longer than in a small volume of water. And if there's energy, infrared energy coming from outside, it will persist longer. Wow. By longer, I'm talking weeks to months, maybe longer, but we haven't measured longer, so we don't know. But solidifying the EZ water is the best, I think, opportunity for making it, allowing it to persist.
Excellent.
I hope that answers your question.
Yes, it does. Because the only thing that we've come up with, and this is through another group that we've been working with plant material and plant growth, is that we were extracting what would be your EZ water out of green, fresh, leafy vegetables by using brown sugar as a way of pulling the water out of those cells. And because that ends up into a syrup, it's very similar to what you were showing in your diagram of the proteins and stuff inside the cell. We can store the liquid for a long period of time. It doesn't change much, but small amounts can be added to with more water to water the plants, and we reduce the amount of fertilizers stuff we need for it. I am so
Brilliant.
excited.
Brilliant. Yeah, that's great. It doesn't surprise me because, you know, sugar is a great structurer of water. You can you know, that's why sugar can form crystals. You just get rid of the excess water, and it's the EZ water that's essentially responsible for the crystallization. So yeah, go to it. I'm afraid I really need to leave.
I know you have to go. We won't keep you, but we would like to invite you to come back to talk at FESIG again. How about the next session in October? Would you be free?
Let's talk about offline, okay?
Yeah, because there's so much questions we'd like to ask you. We've learned so much from you. It's really wonderful to learn more. If we could.
Thank you, Crystal. I appreciate that. Okay, I got to sign off
honor.
for the moment, and please take care. And thank you for the
Professor
invitation.
Thank you so much.
Jerry. Thank
Yeah,
you.
my
You too.
pleasure.
Take care.
Okay, bye.
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 Let's see.
Right. So there
Stop
being
share.
no other business now, this first session of the 92nd FESIG meeting is adjourned to the second session. Thank you. You can stop recording.